Yeah. It's not on, but this switch is right there when you're ready. They're just hanging out by the water.
No, they're like it's an assembly line. They're like fascinating and coming in.
That's why you gotta get here early.
Okay, all right. I'll give them I'm gonna do a. And am I supposed to use this mic or
Yeah.
because that will be better.
You just gotta, you know, the teaching setup here is awesome.
No. I'm supposed to press OK, but it's being live streamed. All right, I see the coffee backlog is in, yeah? All right, so everyone's caffeinated, we're good to go. Well, it's my pleasure to introduce Dr. Kim Komatsu, who's today's seminar speaker. And so Kim is a plant community ecologist. She got her PhD at Yale University working with Dr. Melinda Smith. And then she did her postdoc at UC Berkeley with Dr. Ellen Sims. And she went on from there to have a position at the Smithsonian Institute before moving 3 years ago to the University of North Carolina, Greensboro, where she's a faculty with a fancy title, like, I don't know, some sort of endowed fanciness. Sorry, Kim. But I've been a really big fan of Kim's research for basically my entire ecological career. So Kim, to me, has really inspired me on 3 axes. The first is that she is incredibly adept at data synthesis and using kind of long-term records and pulling together data together creatively to answer really big questions in how communities respond to global change. The second is that she takes a really broad view of species interactions. When our lab wants to think about plant-insect interactions, she's the one we talk to. When our lab thinks about plant-microbe interactions, she's the one we talk to. She really has that kind of broad perspective. And the third is just as an ecological community member, Kim is really committed to kind of open science and training that next generation in working with these large data groups and really mentoring kind of next generation of scientists and taking leadership positions within the ecological community. So I'm a big fan. Every time I feel a little jaded with my work, I look to Kim, I get re-inspired, and I'm so happy to be able to share her with you guys as her speakers today.
Very nice introduction. Thank you, Lauren. I've known Lauren for a long time now. She was an undergraduate student just finishing up at Yale when I started my PhD there in Lee's lab. And then she went to do her PhD with my former undergraduate advisor, Katie Studing, who had moved to Berkeley at that time. So we have a long history of discussion and collaboration, which is really fun. So I'm very glad to be here and thank you all for coming. So Lauren mentioned that I do a lot of data synthesis, and that's what I'm going to be talking about today, some of the data synthesis work that I do. But I just want to start out talking about the ecosystems that I work in, sharing a little bit of my love of grasslands with you so that you can all hopefully be interested enough in grasslands for the next 45 minutes for me to talk to you about that. So I love grasslands for lots of reasons. The first is that they're just beautiful landscapes. So this is one of my field sites out in Kansas at the Concurrent LTDR. And I think hopefully anyone looking at this picture would look at it and say, this is a gorgeous landscape with these big open skies. We have our green grassy fields below in the spring between our burned areas and green, our unburned areas in the tan color. So they're wonderful places to work in and to live in. And humans have been living in grasslands for a very long time. It's thought that grasslands are a driver of human evolution, which is super cool. So it's thought of things like lineages that led to our bipedalism or our complex social structures as humans all arose out of the seafloor grasslands in Africa. So we have grasslands to thank for the way we are today. And then, of course, they're also ecologically important, right? So grasslands cover 40% of Earth's ice-free terrestrial surface. They're home to a wide variety of plants and animals, many of which are endemic to grassland systems. They provide really critical ecosystem functions and services for humans. So things like water filtration and nutrient cycling to produce our fertile soils, lots of below ground carbon storage. So they rival our forests in terms of their carbon storage. And ecologically, they're really highly tractable systems. So these plants stay put. They grow really quickly. They're really responsive to disturbance. So a lot of our big ecological theories have come out of grasslands that then we can translate into our other systems as well. So lots of great reasons to love grasslands. But our grasslands are also endangered around the world. This is that just for North America, that we only have 40% of our original grasslands remaining, and only 2% of those remain intact. So most of our grasslands in our continent are degraded. So not only are they important to think about in terms of their ecological importance and their cultural importance, but also they're important to think about in terms of restoration. So I'll stop my soapbox about grasslands there, but hopefully I've convinced you that they're interesting to think about. And so jumping into the science here, we know that our plant communities and our grassland systems are changing over time. They might be changing in response to environmental drivers or through successional processes. And this is something that I'm really interested in studying. And we also know that humans are actively changing our environment, right? So this figure is showing as the global human population is decreasing, we're seeing concomitant increases in carbon emissions and atmospheric CO2, nitrogen emissions, the global temperature anomaly. So humans are having a big impact on our planet that could be changing these dynamics within our plant communities over time. And we are fortunately also in an era of global change experimentation, right? So over the past several decades, people have started to try and impose global change treatments in an experimental manner in a field setting to figure out how are these plant communities responding to things like drought or fire disturbances or nutrient pollution. What can we say about what's going to happen in the future, right? So this is great. That we're doing this, but what we're finding is that repeated experiments can have really divergent outcomes. So this is just an example of some work that we've done at the Ponza Prairie Long-Term Ecological Research Station. This is a tall grass prairie site in eastern Kansas. It's a beautiful field station. It's where I do most of my research. Most of the background photos in my talk are from Ponza. And here we have been doing science for the past 40 years to try and figure out how this system functions, right? And at the site, we are fortunate to have 6 different long-term experiments that have all been manipulating nitrogen availability over decade-long series. And they're all doing it in the same way. We're adding 10 grams of nitrogen per meter square, and we're tracking the plant change or the plant trajectory over time in these experiments. And so this figure is showing for all of those experiments with the different colored points, how is the plant community responding? So there's a multivariate space where each point represents an entire plant community. So all of the species identities and abundances are encapsulated in that one point. And the further away the points are from each other, that means the plant communities are more dissimilar in terms of their species abundances and species identities. And so what we're seeing here, each of the different colored points represents a different experiment. The head of the arrow represents what the plant community looked like at the beginning of that experiment. And then the tail of the arrow represents what it looked like at the end of the experiment, after at least 10 years of nitrogen addition, but in many cases, many more, up to 30 years. And we're seeing that everything is all over the map here, right? So we are not seeing a consistent response across these experiments to nitrogen addition. They tend to start in different places, and they definitely end up in different places. And I'll just highlight, there's one example here, are teal and darker teal dots here, are 2 experiments that are co-located in the exact same watershed. They were started 1 year apart from each other. And you can see initially they have very similar plant communities, as you would expect for 2 experiments that are literally 20 years apart from each other. And yet, after 10 years of nutrient addition, they end up in very different places, right? So how can we say anything about how plant communities are going to respond to global change if even at a single site with replicated methods, we're not getting consistent responses? And so this is the question that I'm trying to address with the synthesis work that I'm doing. Can we generalize our plant community responses to global change at all? And I'll highlight that most of this work has been a collaborative effort with my science life partner and best friend, Megan Holder. So Megan and I started together in grad school and IE2 Lab, and then have embarked on this collaborative endeavor together over the past 20-ish years now. And so none of this work that I'm showing you today would be possible without her. And I asked her to send me a recent photo of herself for this talk, and that's what she sent me. Not very helpful. So here's an old photo of Megan, so you can get a sense of what she looks like. So back when we were graduate students, Megan and I have been working out at CoRRE, and we saw these patterns, right, of different responses of our plant communities to the different experiments that we were working on. And so we wanted to try and generalize, can we say something globally about how plant communities are responding to global change? And so we were fortunate to receive funding from the LCBR network, and we got together this great group of collaborators who at the time were mostly either grad students or postdocs. We now all moved into faculty positions around the country, which is very fun, to try and tackle this question. And we've continued this work over time with 2 different iterations of funding through NCs and I did, and added in a whole host of other collaborators from around the world. So again, none of this work would be possible without all of this great team of people behind the scenes. Okay, so as part of this working group, we developed the CoRRE database. This is a database of experiments that are all independently run all across the world, where we've gathered the data from the people who are running the experiments. And these experiments had to directly manipulate at least one resource. So they had to either manipulate nutrient availability, water availability, CO2, or light availability. And then they had to be in our tractable herbaceous ecosystems, have at least 5 replicates, and have run for at least 3 years. So we could really see how these communities are changing over time. And so with that criteria, we came up with 134 different experiments from 64 sites around the world. And many of these are long-term data sets, so we have lots and lots of repeated data points from these sites over time. And so our first question here, way back when we started this work, was can we generalize our plant community responses to global change drivers at these sites across the world? And so to do this, we started by looking at a really common metric that people use to characterize plant communities, which is plant species richness. So this is just a count of the number of species that you see in a plot. And so here we're looking on the x-axis at how long the experimental treatment has been going on. And then the y-axis is the proportional difference in richness between our treatment plots and our control plots. So positive numbers mean you're seeing an increase in the number of species in your plots. Negative numbers mean you're seeing the number of species. And then each of these individual gray lines is an individual treatment response at a given experiment in a site around the world. So you can see there's a lot of variability here. And this big black line down the middle, it looks like this axis, but it's actually the global mean. So we're seeing globally, on average, no richness difference through time across these experiments. And even though there is a lot of variability between lines, 78% of our sites show absolutely no difference in plant species richness in response to global change treatments. So we're finding that richness is really not a very sensitive metric to global change treatments. So we can do better. So we started to look at these multivariate metrics instead, similar to the figure that I showed you from concurrent. So we can calculate what's that spatial distance between our treatment and control plots in that kind of multivariate space that takes into account species identities and species abundances, not just the total number of species. And so here we see a very different pattern, right? Our global mean is a linear and positive line. So we're seeing increased plant community change with our global change treatments over time. But again, we have a lot of variability. So sometimes we have individual treatments at a site that might have an increase in these global change, or sorry, an increase in our plant community difference with these global change treatments. Sometimes we see no response, so these flat lines down here. Sometimes we see parabolic responses, so the community initially changes, but then somehow it reverts back towards the control conditions. So we still, even though we're seeing that plant community change is happening, we don't have a very good handle on exactly what is happening. So overall, we're finding our community responses to global change dynamics or global change drivers are very dynamic across these sites. So that's a lot of preamble to get to the main question here. of how can we get closer to generalizing these plant community responses to global change at sites around the world. And back in the late 90s, early 2000s, people had posited that plant traits should be the holy grail, the way that we can characterize what species are going to be winning and what species are going to be losing with global change, and what that should mean to the functioning of our systems. So basically, we're saying we're taking pictures of leaves, among a few other traits, and measuring how broad are the leaves, how thick are the leaves, how much do the leaves weigh. These are all supposed to be things that can help us predict whether these species are going to be succeeding or whether they're going to be failing with global change and what that means for our communities. And so we feel like now we're finally at a point here where we can use plant traits to try and make these kinds of predictions. And so the two questions I'm going to talk to you about today are, can we use plant traits to understand mechanisms of species co-occurrence? And then what traits make species either winners or losers under these global change conditions in our experiments? All right, so I'm going to take a little bit of a tangent here, talking about plant traits. So we have about 6,000 species that we're interested in across our CoRRE database, as well as two other databases that I'll mention in a minute, that we wanted to characterize for their traits. And we were looking at 12 different traits, 6 categorical traits. So things like, what's their growth form? Are they a grass? Are they a forb? Are they woody species? What's their photosynthetic pathway? What's their lifespan? Are they clonally spreading, or are they reproducing only by seeds? Do they have microbial mutualists like mycorrhizae or nitrogen-fixing bacteria? And then we also looked at a bunch of continuous plant traits. So how tall is the plant? How broad are the leaves compared to their weight? How much weight do they have compared to their wet weight? A bunch of nuanced leaf traits, right? But all of these traits are supposed to mean something about whether these plants are acquisitive, are they fast growing in high resource environments, or are they conservative, can they handle stressful environments well? So this was our goal, to collect all of this data for all 6,000 plant species. And when we went out to look at the plant trait data, we found that the trait data are really sparse. They're really messy. They're flawed in many different ways. And so we had a real challenge. So this figure is showing, you don't have to look at all the details, but we're looking at about 5 different plant trait databases, including Tribe, which is like the #1 holy grail plant trait database that all of people use. And we find that for our 6,000 species, when we're looking to see whether they have any data, in most cases, we have maximum about 20% of trait data for these species. In many cases, we have less than 10% of trait data across all these databases. So basically, we have gaps of 80 to 90% in our trait database that need to be filled, right? In addition, this data is often wrong. So you can tell this more easily from categorical traits. If something that you know is a grass is labeled as a forb, you know that's wrong. But how do you know something's wrong when you're given a type version, right? It could be correct, it could be a really tall individual, or it could be not correct. And then a lot of the data is also repeated across these databases, so there's a lot of being involved to get to the point where you can actually use the data. And even when you can use that data, you're missing 90%. So what we did here is we painstakingly, as a group, went through and categorized all 6 of those categorical traits for all 6,000 plant species by hand. So we went through the literature and found for each of those plant species, is it a grass or a forb? Is it clonal or not? And we built in a data sheet for that. That took many person hours. And then for the continuous trait data, we used Bayesian hierarchical imputation methods to figure out what are the relationships between these various different traits. So how is plant editing trait related to nitrogen content and so forth? And what are the phylogenetic relationships among all of these plant species? And then we used computer simulations to impute the data for the 90% of data that we were missing. So basically, we made up this data in a very scientifically robust way to have a complete data set in the end. So there's a lot of work to do, of course, but now we have this incredible database, which I would encourage anyone who's interested in plant traits to use. It's all publicly available on the Environmental Data Initiative repository. So now we can ask our questions, first focusing on whether we can use these plant traits to understand mechanisms of species co-occurrence. And so I'm going to get a little bit into the weeds here for all of our non-plant people out there. So this is what one of my field studies looks like. I go out into the field, I bring my quadrat, I'll lay it down in the field, and I'll assess the plant community composition in the field out there. So this is a picture of what I'm going to be seeing in some of my plots, right? And it might just look like a sea of plants to many people, but I've spent enough time out in these plots that I can say we have some rare species that are present here. We have a white sage, the aster at the top side of the ground here, grasses. These are all the rare species in the system. When we're thinking about our plant species richness metrics, these species, of course, are overweighted in those metrics relative to their abundances, right? But they are important. But the vast majority of the plant species out here are made up of two different species. We have our prairie dropseed, which I've highlighted in purple, and then our little blue stem, highlighted in yellow. Yes. And so these two species are co-dominating in their abundance in this ecosystem. They're each about 50% abundance in the system. And they make up the vast majority of the functioning of this ecosystem, vast majority of the production, right? So co-dominance is a term that's been used for many decades in the literature, but hasn't been well defined in the past. So recently, Jess Ray and myself and Lee Smith have come up with a more holistic definition across all the different papers that we've seen, that co-dominant species are species that have a shared abundance that together comprises the majority of the total abundance. And so that's what I'm going to focus on today, is what's driving our co-dominant species. And why should we even care about co-dominant species? Why am I telling you about this? They should be important for ecosystem stability, right? So they're making up the vast majority of our functioning in our ecosystems. You could think that trade-offs either temporally or spatially in the environmental conditions that might favor one of these co-dominant species over another could potentially allow them to stabilize each other and therefore stabilize the entire ecosystem. They're important for restoration efforts, so we can't just bring one species back. We might want to bring back multiple co-dominant species if they exist in the system. And what I'm most interested in, that is for the purposes of this talk, is that we can use them to say something about coexistency. So how is it possible that we have these two species that are both super highly abundant in the ecosystem? Why don't they outcompete each other? And so that's the question we're asking here with our plant trait data. What filters are influencing species co-dominance within our ecosystems? And to do this, I mentioned we use a couple additional data sets. So we're using the CoRRE data set, but in addition, we added in a bunch of experiments from the Grazing Disclosure Consortium. So this is, again, an assembled database of independent experiments where people are manipulating grazing pressure and abundance within grassland ecosystems. And then we also use data from the nutrient network, which is a coordinated distributed experiment where people are using the same exact methods at hundreds of sites around the world to manipulate nitrogen, phosphorus, potassium, and herbivore pests. So across these two data sets, these three data sets, we have 527 experiments represented in this synthesis. And so what do we see when we're looking across these experiments? What do our dominant structures look like? In most cases, which you can see as these blue dots at our sites all around the world, we have monodominated communities. So there's a single dominant plant species that makes up the vast majority of abundance and functioning in the system, and everything else is relatively rare and relatively even. But in about a quarter of the cases, which is a non-trivial amount, we have co-dominance where we have shared high abundance of 2 or 3 plant species before you see that drop off to the more rare species. And then fewer sites also have even communities where everything is relatively evenly represented. So we see that co-dominance is relatively common, and therefore important to think about, I would argue. And it's especially common in more productive systems. So when we're looking across environmental variables like median precipitation and median temperature, both of which relate to higher productivity grassland systems, we're seeing that monodominance declines as we get to these higher productivity systems, and co-dominance increases, right? To the point where the probability of finding a site that's co-dominated is greater than finding a site that's monodominated. So it is important that these higher productivity systems for sure. And so we can ask what's driving co-dominance, co-occurrence within these systems, and you can think about this from a standard community assembly framework. So we might imagine that we have our species pool at the top there represented by all these fun little shapes. And those species might arrive in a location where they encounter some sort of environment, right? And can they tolerate those conditions? Can they persist there with the amount of soil moisture or temperature that they're experiencing? That's our first abiotic. And we might expect that this environmental filtering or this abiotic filter could promote codominant species that have similar traits to each other, right? They're being constrained by the environment to have a certain set of traits or trait syndrome. And we might expect this to be more common in more stressful environments. And we can compare that to our second filter here, our biotic filter, where once the species can persist in the ecosystem, then they will start interacting, right? Out with each other. And so in this case, the biotic filter could be filtering for species that have trade-offs along their trait axes or more dissimilar traits from each other to promote their codominance through niche differentiation. And we might expect this to be the case in more resource-rich environments. And so we're asking what filters are driving species codominance here. So this is a trait dendrogram. It looks like a phylogeny, but these are actually trait distances across our different species that are incorporating all 12 of those categorical and continuous traits that I told you about before. And we can look at any pairwise combination of species and say how dissimilar are they in their traits from each other. So these would be very similar. These two way across the way would be very dissimilar from each other, right? And so we can come up with a null distribution across all possible pairwise combinations of species within our environment. And you might expect to see a hump-shaped curve or a bell curve of trait dissimilarity across all of these species as our null expectation. And then we can say, how does our codominant pair? So imagine we have these two species are our codominant and everyone else is rare in the community. How does that codominant pair compare in their trait similarity to everybody else? And if they have relatively similar traits, low trait dissimilarity, we might expect that to be indicative of that environmental filtering, those stressful conditions. If they have relatively dissimilar traits from each other, potentially they're experiencing niche differentiation, right? All right, so now we can look at across all of our sites that have codominant species pairs. What is that relative deviation in functional distance between the codominant pairs relative to that background null distribution? And so here we're seeing a bimodal curve. We have a lot of codominant pairs that have more similar traits to each other than would be expected from the null distribution. So again, indicative of that environmental filtering. But we have the second peak over here, a non-trivial peak as well, that have more dissimilar traits from each other than would be expected by chance. So again, indicative of that niche differentiation. So we're seeing across all of these sites, either environmental filtering or niche differentiation, depending on the site, is promoting codominance in these systems. And that's under ambient conditions. What happens if we alter the environmental conditions? So we should be driving species shifts if we're altering our global change conditions, and potentially altering our filters as well, right? So you might induce more stressful conditions that could increase that environmental filtering, or you might be inducing more competitive conditions by increasing resource availability. And so we would predict with global change, we might see a decrease in codominance as we're forcing these species into or out of the boxes that they've been living in. And certainly we would expect to see shifting trait dissimilarity with these global change treatments. So is this the case? So first here we're looking at a safety diagram that's showing what is the trait syndrome across our various different treatment types. So the left-hand side here is showing under ambient controlled conditions. And as I mentioned before, we have a lot of nominated sites. But a fair number of codominant sites in the red here and some key sites in gold. And then how do those conditions change when we apply our global change? And we see the opposite of what we predict, if anything, an increase in the incidence of codominance with our global change treatments. But where those trait syndromes are coming from, or those dominant syndromes are coming from, is really all over. So a fair proportion of the previously codominated sites become monodominated, or even some stay codominated. Same thing with the monodominated ones. It seems to be about proportional, the ones that stay monodominated versus switching to co-dominated or even. So really all over the board in terms of what turns into what with these global change. So not what we expected. But then when we're looking at our filters and asking whether our filters are changed with global change, we do see patterns similar to what we would expect. So just to remind you here, this is our control conditions where we saw a peak of co-dominated species that have environmental filtering signals, as well as a second smaller peak that seems to have more dissimilar traits, indicative of niche differentiation. When we apply global changes that are stressful, so things like drought or warming or intense grazing, we see a shift towards species that have more similar traits. So those co-dominated species have to be more similar to each other to tolerate these more stressful environmental conditions. And on the other side, when we impose global change treatments that increase resource availability, like irrigation or nutrient additions, we see a shift in our peaks for more dissimilar traits, right? So species are separating out in their niche space to try and make a home for themselves within these new conditions. So overall, we're finding that both these abiotic and these biotic filters are maintaining co-dominance in these ecosystems. The co-dominant species tend to be more functionally similar under ambient conditions than we would expect by chance. So it seems like we're seeing signals of environmental filtering across a lot of these systems. But there are a substantial subset that show trait divergence among the co-dominant species as well. And then global change is definitely shifting these dynamics. Stressful global change drivers are increasing or strengthening that environmental filtering, whereas increasing resource availability is keeping differentiation in these systems. All right. So we can definitely now use these plant traits to understand how are plant species co-occurring in our ecosystems. We know that these communities are changing with global change. So now we can ask what traits are making species winners or losers under these global change conditions. All right, so to do this, we use another metric of dominance of species within ecosystems. So we use the dominance candidate index. I'll just call it the dominance index for this top. And so the dominance index is thinking about the abundance of species within our plots as well as their frequency of occurrence within our plots. So a species that is maybe actually highly abundant, but is not very frequently found across the environment would have a low dominance index. And a species that's found within many plots across our ecosystem and at relatively high abundance would have a high dominance index. And then we can compare these DCI values of species in our control plots versus our treated plots. So if they are more abundant and frequent in the treated plots than control plots, then we would call them winners with global change. If they're more abundant in control plots, less abundant in treated plots, we would call them losers with global change. And so then we want to ask what traits make these species winners or losers with global change? Why are we having this distribution of some species winning and some species? And this is something that people have been thinking about for a long time, of course. So thinking all the way back to Brian and the CSR triangle here, where we predict that certain species might do well in their highly competitive environments, other species might do well in stressful environments, others might do well in high disturbance environments. And there's a fun recent paper that just came out where people use plant traits to map onto this triangle and say that things, for example, like high mycorrhizal colonization leads to better competitive success of species within our environments. Or higher growth rates or more acquisitive traits could lead to greater success in high disturbance environments, right? So we can directly map our traits onto these ideas of who should be winning and losing in different environmental conditions. And we might expect, as you would think from this figure, that specific trait syndromes should be important under different environmental conditions or different global change drivers. So nutrient additions, you might expect having high specific leaf area or tall plants. So again, these species that can grow really fast and take advantage of the resources would be very useful. In high disturbance environments, you might expect having low seed mass could be good, so you could disperse into these areas. Areas where they're being warmed or droughted could benefit from having a C4 or a CAM photosynthetic pathway. that these plants tend to be more water efficient in their photosynthesis. So you can make predictions all the way down the line for all of our different traits and all of our different global change environments. And so we tested these predictions, many of which I, of course, am not showing you today, using our CoRRE database, thinking about the winners and losers of global change. And so here, these figures that I'm going to show our different traits that are different panels. Each of the bars represents a different global change treatment. And then the x-axis is showing the effect size of that difference in the DCI value by trait. So basically, you're a winner. You're a winner with a given trait if you're on the right-hand side of this graph, and you're a loser if you have that on the left-hand side of this graph. And so we could use our categorical traits as kind of like a gut check to see, are we seeing the patterns that we would expect to see here in these ecosystems? And we definitely do. So for example, this top graph here is looking at whether being a C4 or a CAM photosynthetic pathway is beneficial. And we see that those plants that have those photosynthetic pathways decline with irrigation. So being really water efficient means that you're not going to do well if you start suddenly having a lot of water in your environment, right? And outcompeted by all of those species. Similarly, if you're a nitrogen fixer, so you associate with nitrogen fixing bacteria, you're probably going to lose. And our data shows that that's the case across all of these data sets, all of these experiments, that the nitrogen fixers are declining when we add nitrogen to the system or if you add multiple nutrients to the system. So it does seem to work with our intuitive predictions. And then we can start to ask a little more complicated questions. So we can think about these leaf traits that people have assigned to either be acquisitive traits, so fast-growing species have high SLA or high leaf nitrogen. And those species tend to do really well if you're increasing nutrient availability within our systems. So you're increasing resources, the fast-growing plants can take advantage of it. And in contrast, species that have low leaf aromatic content, LVOC, are thought to be more resource conservative, and therefore they should be doing better under low resource conditions, right? But might be doing more poorly under resource conditions, and we can see that in the case as well. So overall, we can use this data to predict who are the winners and who are the losers of global change based on the traits that these species have. All right. So just to sum up what I've talked about here today. So we're synthesizing across hundreds of different experiments. And we do find some broad generalizations, which is a step forward from where we were 20 years ago when we started this work. So we're finding that plant communities are definitely changing. But species richness doesn't seem to be a sensitive metric that we should be using to predict or to detect these community responses to global change drivers. We have to be thinking more holistically about our plant communities, who the species are that are present, and what their relative abundances are to each other. And then we should also be thinking, as was expected, again, about 20 years ago, 30 years ago at this point, that we can be using these plant traits to try and make predictions about how these communities are going to change through time with these global change treatments. So we can use plant traits to understand what are the mechanisms of species co-occurrence under these ambient and global change conditions. And then we can also use these plant traits to predict which species are going to win or which species are going to lose with various different global change drivers. All right, so I'll just wrap up here with the last 5 minutes or so that I've got left to think about where we should be going from here. So this work is all definitely on the theoretical and statistical model side of what we've been doing in community ecology. So trying to predict resource use, stress tolerance, think about competitive exclusion in these ecosystems with our traits. Thinking about beneficial interactions between species, so our mutualistic symbiosis in systems, as well as species' ability to disperse and their demographics, their plasticity, right? So we can use our organismal traits to predict our community structure based on all of these effective models. We can also use traits at a muted weighted trait level, which I didn't talk about today, or we're working on, as well as thinking about, again, those competitive and beneficial interactions to then make predictions about how organismal traits should be expressed for our specific environment. We can think about is ecophysiology of our plants and what that means to the functioning of our ecosystems. And we can think about competitive and beneficial interactions, trophic dynamics, niche partitioning, and our dominant species. To think about how our plant communities and how they're structured are influencing functional partnerships. So these are all things that we're doing really well with these big data synthesis efforts. We're making a lot of progress. But then on the other side of things, we have a bunch of process-based models that are trying to make much broader predictions about how our ecosystems are functioning, right? So they're doing this at a regional landscape scale or even a global scale to say how are our ecosystems functioning in terms of overall production or a variety of other services that ecosystems provide. And so they're thinking much more about competition among species or competition among functional groups. They don't even get down to the species level in most cases. And specifically, they're thinking about carbon acquisition and how these species are limited by light availability or water availability or nutrient availability. Those are the traits that they care about. Those are not the traits that we're measuring on the ground. Right? They're thinking a lot about variation among preset plant functional types. So they take a plant functional type, they'll parameterize it, and then they'll put it in their model and interact with other plant functional types. And that's the mediated structure that they're and they're doing this to say something about fluxes in the environment, right? And then ultimately, they're using the plant traits to predict these fluxes of water and CO2 and energy in the environmental systems. So we're really thinking on, I would say, two very different scales. We're measuring things on two very different scales. And we have a really hard time talking to each other. And so a big part of the work that we've been doing is bringing together these empiricists who are on the ground collecting our plant trait data. We're like measuring our weeds and thinking this is so important. And this is not at all what the ecosystem modelers care about. And vice versa, they're incorporating mechanisms into their models that we might not on the ground be thinking are as important as empiricists, right? And so we need to be interfacing more and talking to each other, finding a shared language to figure out how we can incorporate the most important things from both sides of these things into both of our thinking, so that we can be collecting the right kind of data to make sure that they can incorporate them into their models and vice versa. And making progress together as a field. So this is, I would say, the last big piece of what we're doing with the core working group is trying to figure out how can we all be working together to improve these ecosystem models or process-based models to make predictions about how our ecosystems are changing in the future. All right, so that's what I've got. Thank you all for listening, and I'll thank our funding sources. And again, I'll just mention, of course, none of this work would be possible without the many, many collaborators that were involved in the work as well. And I'm happy to take questions.
I was going to ask about why all of this unexpectedly done at more they should be focused on these maybe students. So I guess I was thinking like if the model dominates these really less well, that kind of creates an opportunity for other species like so that kind of makes sense to me that it makes
Yeah, I feel like I could see it either way. I could imagine that there would be cases where if the monodominant species is a loser, right? It's being lost from the community, then that maybe gives the opportunity for multiple other species to kind of vie for that new role. So you might see an increase in codominance initially, but my thought was that through time, you would expect that whoever was coming into, that new position would outcompete anything else, right? It would be the ultimate winner. And so we would see a decrease in the incidence of codominance just because of those species interactions that might be strengthened with these global change drivers. Maybe not as many species can tolerate the new conditions, or maybe just the strongest competitor comes in and survives. But of course, you could see it in the opposite direction as well. And clearly we did, right? And what's interesting about that data that I didn't show is that when you have these co-dominating species under global change periods, It's really 50-50 whether they're the same species that were co-dominating or if they were originally monodominated. Was that monodominant species still present in the co-dominant pair? About half the time that was the case. The other half the time, it's completely new species that came in. So what's happening there? I have no idea. But it's a really interesting pattern that we're hoping to follow up on to say, is there some sort of trend that we can look at for why certain species are doing well in these situations? Yeah. Yeah.
Are the monodominant species the same before?
We didn't look at that, but that's a good question. My guess is it would probably also be about a 50-50 split. That some would be the same and some would be different.
I have two questions about the predominance thing. The first, it's great to see this scale of the data. Really fascinating. The first question was about the functional trait divergence method that you're using. So you do this in the context of species that are related on phylogenetic. It's already controlling for the phylogenetic relationship among morphological traits.
We are not controlling for that. No, so we're just looking at the trait dissimilarity and not the functional for the biotic. But I will say, as there's been like 24 different projects as part of all of this work, and we are certainly looking at phylogenetic as well. Is there a phylogenetic signal? Who's winning? Who's losing? Is there a phylogenetic pattern in terms of functional trait dispersion versus phylogenetic dispersion, and even responsible looking at the community level? So it's something we're considering, but yeah.
Yeah, it seems like that would be super important to expect. Oh, these are congeners and have similar trait that could just be biological.
Yeah.
So then my second question is with respect to environmental filtering. I was kind of surprised at what you said, because so if I got it straight, it seemed like you were saying that environmental filtering under smaller stress conditions should lead to predominance. But the data that you showed that with very low like precipitation, those were like bottom kind of
Yeah, we're seeing different patterns in response to the global change signals than we do with the global understanding. So I would flip around the statement that you said and say, we're looking at predominance as a way to test these filters. And then in the predominant species, we would expect that stronger environmental filtering would be under more stress conditions, and stronger trait differentiation or differentiation would be under more stress conditions. And we find that with the global change treatments, the stressful treatments led to more trait similarity, and the resource treatments led to more trait dissimilarity. But then at the global scale, the more productive sites tend to be the ones that have greater incidence of predominance. So it's kind of parsing the question in two different ways. Where do we see predominance globally? And then once we have predominance, how is it responding?
I have somewhat uncomfortable questions, but I was curious about. So it seems like a lot of the treatments that you guys are looking at the database are like adding nutrients or adding some extra for the plants, and I maybe not usually I kind of associate with the global climate change stuff and feeling that that's like taking away those things. And so I was curious like how much your results can be flipped around to answer questions about like lack of treatments.
Yeah, I feel like we could make the opposite prediction, right? That if you take away resources, then you should see more stressful environments for these plants, and they should have a certain suite of traits that would allow them to adapt to those conditions. Yeah, with global change, I would say we're much more frequently seeing increased nutrient availability specifically, right, through like air pollution or runoff from agricultural fields. We're putting a lot of nutrients into our ecosystems. So in that case, we're seeing increases, but of course, with climate change, we're seeing things like increased drought incidents, right? And so that is taking away resources. And we categorize drought under our stressful treatments there as well.
Yeah, I have the patterns both kind of across systems and the patterns of like having more the similarity of adenosources and convergence once you lower the sense. And I was wondering if you then maybe coordinated responses in relation to the kind of cross-site axis, like is there some sort of threshold with MAT or if you drought and you get pushed to the dominant state, or some sort of threshold with MAT where you add that nitrogen, so it's enough of an axis to differentiate. Does that make sense?
Yeah, it makes sense. And we did do this and we saw no clearly discernible pattern to say at higher resource sites or at lower resource sites in terms of their global environmental conditions. You saw one pattern or the other in response to global change. We also tried to look, so I showed that binodal distribution of trait dissimilarity across our control plots, right? And we tried to look to see, is that controlled by the environment that these systems are in? Can we say that the ones that were the peak with the environmental filtering are the lower resource sites, and the ones that are the peak with the niche differentiation were the higher resource sites, and there was no pattern there either. Way cool, but
Great. I'm curious about two things. At least two things. First, how do you handle pretty curious to see the data spread and the data shifts that then converge back to control. There seems to be an important control on one of this. And each experiment that was planned decade, that's a year that at least 3 years. First question is how are you thinking about managing the control of the ratio of the experiments to this fact? With all of the change, things and how did it be you to see that in treatment of the shit? That's the first question. The second one is how did the ecological community receive the data science approach for the environment or the atmosphere in the recent grades? Was that an uphill battle as imagined was, or is it a hypothetical thing?
yeah. I'll start with the second question. So thinking about the gap filling, we have 90% or 88% missing trait data in this gap fill. And I will say that in our group of collaborators, about 10 people, we spent hours arguing about, is this appropriate? Can you say like the maybe not 90% of the yeah, but we were really confident in the person who's doing the information work for us, and wrote has done this and published this method. She's done it on much smaller data sets. But then when we actually tried it, we found that there were certain traits that we couldn't include because they were way too sparse. So you have like 1% of the data, even if you're looking at these relationships on traits or how the method for relationships on species. It was just too little data to be able to confident. But if we had about 5 to 20% of the data for any individual trait, we did data validation, held back the data, we ran it and we tested the computer results versus the data and held back, and we had really strong correlation. So we were pretty confident in the answer. When that was just within our own group, then when we get excited to publish, it was the same an iterative process of improvements, where again people were super skeptical about this. And good. And then your other question was temporal. Yeah, so we control that on different ways in this data set as well, throughout all of the products that we've been working on. So sometimes we're looking explicitly at a temporal term. Sometimes we take the last year data. Sometimes we take the 10-year data for a data set that has that consistent number. And we see pretty similar responses across all of those different methods. So they were pretty confident in the overall mature see for how our companies are doing. But of course, this really speaks to the importance of long-term data. Because if you only look at the first 3 years, you might see a totally different pattern than if you looked at the 10-year or the 20-year or the 50-year. And what we don't know, so you mentioned those parabolic paths, right? Where sometimes you see increasing community change and then it reverts back to those people.
It's not also the urbanization effect.
Yeah, totally. Yeah. So it could be the case that you're seeing that response to climate. But the controls are also changing through time, right? So it could be the case that the control plots are also experiencing global change And they might also be shifting in the direction of response, which some of our projects have done that's happening as well, but the control plots are actually sometimes changier than the response. And so that's not just a quick response. So really explicitly incorporating that temporal pattern, and doing these long experiments.
And so, like at the end, you were highlighting some of the traits you identified that made sense under the global change regime side of the right? So I guess my short question was, were there any traits that surprised you of like you would understand why they would be associated with that same shift, or like maybe even opposite of what you expected?
Yeah, there were, I would say for the traits, it was all pretty consistent with what you would expect. Some things were much more nuanced than want to share in a talk here, but the traits made sense. But we did the same thing with phylogenetic signals. So we were looking to see, are there certain clades that are winning or losing with global change? And some of those were surprising. So like the Fabaceae losing with nitrogen makes perfect sense, and that came out. Or the grasses doing well under drought conditions. That makes sense. They're very water efficient. But then, and the spurges also did well. So the euphorbs with drought, that makes sense. But then there was like the amaranths were doing well with nutrients and with drought. There's like a few different things that they did well with. And so what's happening in those groups? Yeah. Yeah. This is all across different experiments. So it's not just like one site was driving the trend. So yeah, there's a lot to dig into with this data. All right. Well, thank you all very much.